is visible.
Yes, yes, sir.
So case of alignment that have tried to look and uh, what would be the structural, what would be the evolutionary in the different proteins, different nucleic acids, and all how the phylogenetic trees can be constructed. So we just to refresh the things that the proteins are generally bound by the CONH peptide bond, where COH and NH2 bond gets bond to it. So the major backbone remains the same with the CONH bond, but only the R, that is a side chain that differs from amino acid to amino acid, and that makes the structural as well as the functional differences of the protein. So the amino acid groups that generally determines, so you can go through this table, is that like glycine, alanine, proline. You know that glycine is the simplest kind of amino acid where R chain is only CH3, where R is doesn't have any, it's only H. So glycine non-reactive in chemical reactions, then is the alanine, then is the proline. Then where it acts as what is the main functional features like cysteine, serine, threonine, they are serving as the post-translational modification site. So mainly you can get the serine phosphorylation site is there. So then large and polar is there, glutamine, asparagine, participation in hydrogen bonding or in enzyme active sites, and some basic amino acids like arginine, lysine, histidine, globular protein salt bridges, the glutamate, aspartate, acidic amino acids, or the chain amino acids. So what are their functions? So another thing is that the proteins first, as you all know, that it contains of four structure: primary structure, then it will be the secondary structure, then will be the tertiary structure. There's a globular that on the basis on the packaging of the secondary structures, and finally the quaternary structure of different subunits. So the bond, the primary amino chain is the sequence only. So here you can see that this C bond is this is called the C alpha carbon, and here it contains two bonds. So C alpha C is called the psi bond, and N C alpha N is called the phi bond. So this is plays a major role in determining the these two angles play a major role in determining the structural stability. So major two secondary structures that are generally found is the alpha helix, which is the interaction between the ith and I plus fourth residue of the protein, and which has 2.6 amino acids per turn, or the beta sheets, which are generally aligned and in the planar region with the hydrogen bonds it gets attached. So here now this determination, if you know a protein sequence and you want to determine the protein structure, it is quite important to understand that whether this protein is will be in which shape and structure. So you all might know that this is determined by the generally that the orientation, that how much C alpha C bond or the C alpha N bond can be allowed in a particular region is determined, has been done by the Ramachandran plot, where this is the allowed region and the others are the disallowed regions. So 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 now the thing is that the protein structure database. So PDB is we all know that we have already discussed it before, that the PDB contains the majority of the database that contains the structure. So whatever the structures that are determined by X-ray crystallography or NMR or electron microscopy, their characteristic is they are deposited in the PDB, protein data bank. But now this protein data bank, which we download in the PDB format, it is also a format is there to understand, so that the different software and program can understand. So by the time now, you all can appreciate that why the formats are very much necessary in the context, because we need to use multiple tools, or multiple tools helps in understanding of the different kinds of analysis. So it is quite important to know and understand the different types of format. So this is a simple text file. So PDB format, if we say that download in PDB format, it means nothing, that it means a text file which contains a header of that, which is that. Then these all the PDB IDs contains a full letter code, the title of what is the protein's name, the source of the organism, which from where it has been extracted or from where it is found this protein, the atom number, atoms, and the heteroatoms, which means the cofactors that is remains attached to the protein, or different ligands that remain attached to the protein. Atom means the backbone of the protein structure, the amino acids that are present in the protein. Then atom number 21, 22, 23. You can say atom number, atom name, the residue name, histidine or leucine, and FAD is for the heteroatom. Then polypeptide chain identifier, that how many chains are there. Then three, within the residue number. Then the XYZ coordinate, that in XYZ plane, what is the coordinates of that, and this occupancy and the temperature factor and the atom number type. So this is what we get in a PDB format. So let us now download one and try to find out that we can find out or not. If we go to PDB, and let me check if I have some PDB files. So, you all can open it, right? PDB file. PDB, this, yeah, yeah, Uma Mystery.
Yes, sir, we opened.
Yeah, so I found this one PDB ID, PDB file. So you can see that it is given the header title, then the source. It's not, this is actually a little bit modified chain. This is actually energy minimization and other things have been performed for further analysis. That's why it is showing like this. But here, from here, you can see that whatever you mentioned, like the atom, the type, the atomic number, the unit, and temperature, and the atom type. Okay, so I don't know why this it is taking time to load. So, okay, so another format is the MMCI format. So PDB format is that only it contains header, it doesn't contain so many information. So another format is this MMCIF format where it has also type of it is type of advanced type of format which also contains all these values. So now this protein structure, if you after downloading this PDB files, if you want to visualize, you can visualize in many in this three four types of frames. First is the wire frame type, where you can see. Another is the ball and stick. Another is the space filling spheres. Another is the ribbons. So these are the different kinds of things. If in the visualization software, you can visualize it accordingly. So it depends upon how you want to represent your data or what you need to see. For instance, if you want to see any distance, bond angles, then this representation would help you to understanding the different bonds and angles, different attached to it. Similarly, for the represent of if you want to after predicting a secondary structure, you want to see how the helix and how the sheets are there, then you can able to find this using these ribbons. So these are different kinds of protein structure visualization. So let us look at one of the software that is the desktop where you can visualize the software. Uh, if you all go here desktop, you can find here and actually it need to download it and then use. So I'll just put up in the chat box. So it was previously RAS small was also there. From RAS small, it has now advanced play. It has been RAS top, which is helps in visualization. Then, they want to show fully from download after the formats and all. No, it's not opening. So if I open Raspberry here, so you will after downloading and unzipping, you will get, you can open the application. So here you have to go for new. So if you open .pdb, so just I'm just giving you, this is one which was present. So you can just open it. Then you have the option of rotating. You can rotate along the x-axis, or you can rotate along the y-axis, or you can rotate along the z-axis. So this becomes important when you try to visualize the structure. Means compare two different structures. Then if you want to have make it we try to visualize it or try to compare it visually, so new we need to present in the same kind of uh, same place, same near about similar axis and all. So here you can rotate it, you can spin also. Second. So you can then you can change the visual effects. Then if you want to see the molecules information, okay, so this is the molecular name, number of atoms and number of bonds, everything. You will get the data about this particular structure. Then if you want to see the sequence of this protein, so this is the sequence. So phi psi angles, if you want to find out the phi psi angles, so what is the phi? If you want to see Ramachandran, so this is the different models. The phi psi, the allowed regions, disallowed regions. So it is mentioned there. Now the H bonds, if you want to display, so you can see that their hydrogen bonds will get displayed here. Similarly, you can remove also, remove hydrogen bonds. Now, if you want to see cartoons, it will different kind of visualization. The surface you can check display dots, or you can see that the surface kind of structure you can able to find. atoms labels. So you can see, you can visualize any PDB structure. So if you can download any PDBs, I don't know, I still. Otherwise, you can download any kind of PDB structure and can download the software and can play with it and can identify the pi-psi angles, the sequences, can look into it. So it's a molecular visualization software. So another, so after this is another protein structure visualization software is there. It is the Swiss PDB Viewer. So here also we can view and we can identify. So this also needs to be get downloaded. So you can download from here. This Swiss PDB Viewer. Let's open. So you can see that here it is loaded. Now similarly you can select all residues. You can build. Actually, this is used for also different types of uh, means analysis means performing some analysis before taking these protein structures for further steps like adding uh, residues, adding hydrogen bond, energy minimization, and all other applications that are generally required for the different processes like docking and all. So here you can perform like this, add hydrogens, add the H2O and the tools. So but you also you can detect the domains. Then you can compute H bonds, detect secondary structure. This is we are giving, so it can so you can perform. Here you can see that energy minimization for one compound, compute energy, force fields. These are all required applications when you would be performing docking and all. So here you need to identify all this and to perform all this. So here, from here you can do all these things. Okay, so if these are all when it is when you are trying to do from the sequences and predict structure, then it all needed to fit the model or with, predict the model and how much it is required for that time it is being treated. So Swiss PDB Viewer is generally mostly used for some much more advanced applications, but for if you want to just visualize, also Chimera is there, PyMon is there, so you can visualize all your protein structures from there. So here you can see the Swiss model. For example, if you don't have a PDB structure present in your PDB, means, that protein structure is not present, but you are having in your study an important protein that you can say that you want to identify a drug for that protein and all. So you can load raw amino acid sequence from here and can, with the help of Swiss with model homology modeling, you can build up that structure based on the sequences matching homology modeling or ab initio based modeling. So here it generally uses homology based modeling, and you can build up the structure and can then look for the compute its secondary structure, detect its identify how much it is in the region of that allowed region or the disallowed region, uh, or so you can predict those structures here. So not only it helps in visualization tool, but also helps in build model building and the protein analysis of different kinds of for further analysis. So it is a Swiss PDB viewer. So after this protein structure, now you need to, if you have two structures, then you would also like to compare, right? Compare different kinds of protein structures. So so for that, I told you that rotating is very much required if you want to try to visualize it or try to identify it, compare it visually. So you need to compare it into same. So for this is the first step. Just think that this is the two proteins. Now you are getting this is another protein. Now you need to rotate and come into the same plane, and then only you can compare it. And mathematically, you can say that generally RMSD values are calculated, which are the distance between the atomic distances, the deviation from those for comparing the two sequences, two structures, that how much they are similar or how much they are different. Correct. So now another statistically comparison tools are there is DALI. So you can compare two PDB-ID structures, do two protein structures. So you can identify how much. If you go, DALI is a web server, so you just can use here right now. So DALI server, you can see that it is a network service for comparing protein structures 3D. So if you submit the coordinates in DALI, compare them with protein data bank, may reveal biologically interesting similar detectable by comparing sequences. So you can, if you go for pairwise sequence search, so we need to upload these PDB files or PDB identifier. So if you all are able to open the PDB site, can you tell me of some PDB IDs? Any one of you?
Able to open, sir, but which for which protein? Could you give us an example?
Could give for hemoglobin and myoglobin. Let me just let us take this only. So if we go here, let us complete this test. So here we will download files. So you can see that here's the faster sequence you can download and also the MMCI format, PDB format. So let us download in the PDB format. So if you open here, so now we will upload it.
ஒன்னு நல்லா எங்க பென்சில் கொடு, எங்க இதெல்லாம் அழிச்சு அப்படி ஏன் எழுத மாட்டதெல்லாம்? பென்சில் இங்க எழுதவே இல்லை, இதெல்லாம் எழுதாம பண்ணிட்டான். இது ஏன் நம்பர் எக்ஸ்பேண்ட் ஃபார்ம்ல என்ன பண்றான் ஒண்ணுமே புரிய மாட்டேது. ஏதோ எழுதி அப்படியே சொன்னா வேர்ட்ஸ்ல இல்லையா? அந்த கேலண்டர்ல ஓபன் பண்ணி மேக்ஸிமம் என்ன சொன்னான்னு பாரு முடியுமா? நேத்து தான். இது மாத்து வந்து இந்த ஸ்கிரீன் இப்படி தொங்க வரது கிளிக் பண்ணவே விட மாட்டேங்குது. இப்படி
Sir, your screen is not visible.
Yeah, I'm resharing it once again.
Yeah, okay.
It's visible now.
Yeah, now it is.
Okay. So here you can see if we do pairwise structure comparison. So what we used to do is it alignment comparison and all. Now we can compare its structure also. So here first we have chosen we have uploaded 2 files, PDB files. Now we need to submit it. Okay, so here is this pairwise structured alignment. So each never here you can see that how it is. It's invisible, right?
Yes. Yes, sir.
Yes, see, so if in this both structure, so this is the point where it is similarities found, if you can find out. So here it is marked as this. So C alpha trace is this. You can find out the cartoon way also and can find look at by spinning as well. So you can show the high structures of the different queries. If you go for the sequence wise, you can find out. And if you can see that according to the amino acids where it is the loop, where it is helix, so where it has match is there, where is it conserved, you can find out everything. So you can see that here, if you read here, you can identify that complete sequence. Expand gap option is also there here. Expand gaps because of long, if it is ungapped sequences are there, and the most frequent amino acid is also colored differently. So this is how loop helix is there, and you can also find out in this. So here you can use for comparing. And the another thing important that DALI server gives is the statistical value, and it is this. It is generally given by the Z-score. So Z-score is that the ultimate statistical value that helps identify that how much is your sequences are similar structurally. So based on this Z-score, you can uh, provide or uh, you can conclude about the structural similarity. Correct. So now going another is that the protein structure classification database. So after you have performed the comparison and you have visualized, so the protein structure also can be classified. So here is that scope database is there, which can classify uh, the different kinds of classes and families it belong. So, So it will help us to identify the evolutionary relationship based on the structure. So you can produce by the structural class alpha and beta protein, small proteins, or the protein type, globular proteins, membrane proteins, or you can hide the PDB IDs as well. So if we, what PDB ID we had used here, that HDA. So it doesn't contain, explicitly it doesn't contain. Let us go for, if we see that this is a globular protein, so you will get the overall classes, the structures and different kinds of proteins. So like ferrotoxin, like it is a globular protein. This is the scope ID you will get. This is the fold ferrotoxin like, and scope ID is this. If we go other way around, yeah, so it is a ferrotoxin like and its superfamilies. The different gamma chain, this is the superfamily type. You can understand the different kinds of beta, alpha, beta, two layers of anti-beta parallel sheet is there, the fold. So you can identify, and the structural classes belongs to alpha and beta proteins. So ultimately, you can understand the different kinds classes of the protein structure for this particular protein. That it belongs to the alpha and beta protein. So if it is globular or what kind of protein, you can understand from this database. So another database is there, which is also it's the cat name is the cat database. So for the scope data, if you were to link, yes, see, if you have, if you find out what 3D structure your protein will adapt, protein function, protein evaluation, you can find out all this. So if we if we go by sequence and if we paste it, we can identify. So let us try it out. You can see a type of prediction of an unknown protein sequence. So also, if you know some protein that has a particular domain or particular function and particular structure, and if you want to identify that the known protein sequence will work on it or will bind to it or have the same domain or not, so you can perform this. So just copy paste the sequence. So now we can search it. So cat structural domains it will search in this database, and also the cat functional families. So it will give the result of both the structural and the functional families of the particular sequence we have given. So it is telling that it has found 250 matches. So e-value, with the e-value, you can compare the sequences. So the lower the e-value, the more the description is mainly alpha globin like, and PDB structure is it is matching with 1FTH. So the sequence that we have given it is matching with 1FTH. So that is of the PDB this different from where it has been found and who has loaded and done it. So cat cluster superfamily is globins, functional family is hemoglobin subunit beta. So we have given the sequence from hemoglobin subunit beta only. So you can see that it has predicted, it has matched with that. And the uniprot entries also it has mentioned about the HbA human from where we have. So mostly it is matching with the hemoglobin subunit alpha. So this is the protein structure. So view domain only you can do, or you can view domain in PDB also you can do. So 1FTH is that. So this is, and if we found out the functional families, so hemoglobin subunit beta, it is matching regions. You can see that it is nearly it is matching because it is about the same we are given. So it is the matching functional families. So one is will give you the functional structural classes, another will give you the about the functional families. So this is the representative abdomen. So here you can find out the geo findings. So geo says that the molecule geo contains will give you let you know about three things. One is the molecular function, one is the biological process, and one is the cellular component. So molecular function it can be protein binding. We know that hemoglobin helps in oxygen binding, so it has predicted, and its oxygen carrier activity and all those. So biological process is helping in this blood coagulation or protein neutralization receptor mediate to oxygen transport. So you can find out that it has predicted mostly it is correct, and the where it the protein's localization it is in cytosol, it is in blood microparticle. So most probably it has done correct observation. So this is for the cat searching through sequences. Now, if you want to know about protein evolution, Now the protein function. So the same thing if we can try to identify the protein function. So we can search by sequence also, we can search by structure also. If we give the PDB ID structure, then also we can find in here. So you'll get the similar results that we got it before. So you can find across this different domains. So if you can try with some other proteins. So if we try to find out, so let us look at the structural domains. So see here you can, it's matching here. So here it is mainly alpha, but it has identified it as a lipoxygenase 1, domain 5. The functional family is lipoxygenase. So you can understand that if you give lipoxygenase type of protein sequence and unknown protein sequence, it can easily let you identify that what kind of is there. And mainly, so if you look at the functional, so you can see that it is where it is matching mostly with the functions and the structural family. So it can help you in classifying the basis of the sequence or basis of the structure. So if you have the structure and not the sequence, then also you can do that or vice versa. So it can help you identify. So if you all have any doubt or otherwise, so this was all for today's with the protein structure visualization and classification. If you all have any doubt, or we can conclude today's session. Yeah, Uma, do you have any doubt?
No, sir. Last 2 weeks, session recording we didn't receive. So, is it possible for to you to send the recording link?
Yeah, okay. I think that is okay. I'll ask that. Why not?
Sir, I have a
I'll ask
question.
it. Yeah,
Sir, in bioinformatics, we use a number of databases, right? And a number
yeah,
of softwares.
yeah.
Sir, what software would you recommend in kind of gene editing in a beginner basis?
Gene editing
Yes, sir.
means can you explain some with some example what you want to do?
For example, sir, while in the last session you did DNA visualization in kind of 3D perspective, we did it in a we did it the 3D experience in a database. Sir, is there any software which does that?
Uh, can you tell which database you are talking about?
So most probably it was I exactly don't remember the name. One second, let me just check. So I hope it is somewhere here. So exactly, I'm very sorry. I just don't remember. The actually the my point was that is there any software which does allow that